The second major event of this afternoon's program is the prestigious annual Sir Arnold Theiler Memorial Lecture. I call on Professor Joseph Chamunowa to introduce the distinguished speaker.
Thank you. Good afternoon and good morning. Prof. Peter Sutovsky is Professor of Animal Science and Obstetrics, Gynecology and Women's Health at the University of Missouri. He is recognized worldwide for his studies of mammalian gametogenesis, fertilization, and embryo development with emphasis on the gamete and zygotic ubiquitin proteasome system, abbreviated UPS. Among other discoveries, he was the first to describe the role of UPS in the regulation of mitochondrial inheritance and introduced the concept of extracellular UPS and its role in sperm maturation and fertilization. In his career, Prof. Sutovsky published over 200 peer-reviewed articles and monograph chapters, was cited 7,350 times in Scopus, H-index 45, and 10,300 times in Google Scholar, H-index 56, I-10 index 152, and featured on over 50 journal and book covers. Recent work focuses on biomarker-based flow cytometric semen analysis, semen nanopurification, sperm genome to phenotype research, and sperm capacitation studies aimed at improving conception rates in the artificial insemination of livestock. This year's Arnold Theiler Memorial Lecture is titled Male Infertility and Semen Evaluation, Andrology in the Age of Precision Medicine and Agriculture. Over to you, Prof. Sutovsky.
Humbled to preserve this year's memorial lecture, and I want to start by thanking you again and the Department of Anatomy for nominating me, and I want to thank Professor Vinny Naidu and Professor Marinda Oosthuizen for selecting me for this very prestigious award. And I have a special thanks to my dear friends, colleagues, and collaborators, your department faculty, Professor John Soley and Dr. Lisette Duplessis, without whom none of this would have been possible. And of course, I want to say hello to all attendees and thank all faculty, staff, and students for attending this event and for making this event possible, including our technical support from VetLink. As I said, I'm very honored to present this special lecture, and by no means would I dare to compare myself and my research career to that of Sir Arnold Theiler. However, I was quite intrigued to find out that we do have some shared background in a way that we both grew up in humble conditions, and our parents encouraged us to love nature and inquire about it. And I was really intrigued by the fact that Sir Theiler actually was very fond of insects and interested in entomology, because as a young boy, I actually started collecting insects myself, and this is still my hobby, and I got to indulge it very nicely 2 years ago when I visited Pretoria, and our friends made a visit to Natural Museum of History possible to see their exquisite insect collection. So, of course, growing up, I changed my interests somewhat, and I ended up becoming a reproductive biology and with special interest in male reproduction, or as we call it, andrology. And within the large field of andrology, focusing on male health and male reproductive health, my specialty is in semen analysis, study of the sperm cell, and fertilization. And the goal of this is to essentially evaluate male fertility in livestock species, but also to work on male infertility in humans. And there are different ways to define fertility, essentially. For the livestock and veterinary medicine, the important definition is the power of reproduction and ability to produce offspring, which is of paramount importance for production. Agriculture. On the human fertility side, it is defined as ability of couple to achieve a pregnancy after 12 months of unprotected intercourse. In humans and in animals, there is a number of factors that affect fertility. But for today's talk, I want to really focus on reproductive performance of livestock as it pertains to veterinary medicine. And that includes reproductive management as well as assisted reproductive technologies, and there is a distinct need for improvement in the field of livestock animal reproduction. As we all know, animal production is a very large industry and is very, very heavily impacted by reproductive performance, and we truly need to improve on this technology if we are to take full benefit of new trends in precision agriculture, such as genomic selection, semen sexing, and also introduction of sires into artificial insemination programs without long-term extensive testing. Specific to your interest, it might be important to point out that assisted reproductive technologies also have a place in wildlife management, fertility preservation, and nature conservation. I also want to point out that working with livestock animals as biomedical models is becoming more and more prevalent because of their genetic similarity to humans and our ability to modify them genetically now in a fairly routine manner. Also, what is very unique about livestock animal models with regard to reproductive biology is that we have very ample fertility records from hundreds, oftentimes thousands of artificial insemination services per sire used in production agriculture, and all that goes back to the notion of one health and one medicine for humans and animals. And livestock animal models, therefore, are now finding place in our effort to develop assisted reproductive therapies for treating human infertility, which has been a prevalent health problem for a long time, but now is even more prevalent as there is more access and more knowledge about infertility treatment. A fairly new notion, though, is that fertility status of a male could be used as a marker of overall health, and here andrology can certainly help and contribute because sperm analysis could really be sort of an early sentinel of somatic disease. That's why in the United States and worldwide efforts are being made to develop non-invasive male fertility tests that might actually reveal important information about males or individuals' general health. But once again, today I want to focus on improving livestock reproductive efficiency, and specifically on how this improvement can be achieved through advances in semen analysis, fertility diagnostics, and also in semen processing and purification for artificial insemination. I like to start my talks or introduce into my talks a little bit of history. So that's what I want to do today as well, if you let me. And I start by pointing out that the oldest spermatozoa known to us are actually 100 million years old, recently discovered, fossilized in amber, and they belonged to this little crustacean. Compared to that, the discovery of the spermatozoon is very recent, when it was first observed by Antonius van Leeuwenhoek and his disciple Ham in around 1677, and this was enabled by van Leeuwenhoek's ability to construct the first microscope. What is intriguing is that this first observation of a spermatozoon has been predated by several centuries by the very first fertility test, with somewhat amusing methodology. This test attributed to Italian female physicist named Trotta of Salerno, essentially asked the man and the woman to, quote unquote, make water in separate bowls filled with cereal, and then observe it for 10 days. And if there were no worms, meaning insect larvae appearing in either bowl, that would be a good sign as to that couple's ability to have children. Of course, we have made a lot of progress in fertility testing since then, and the andrology field, as we know it today, became to take shape in late 19th and early 20th century. An example of this is from my own private collection of old biology and medicine books. This Common Sense Medical Advisor in plain English, published in 1909, has a fairly good description of andrological analysis. And once again, technological advances in constructing microscopes have contributed to better semen analysis and better ability to evaluate fertility. So our grandfather andrologists at that time already were able to diagnose teratospermia, essentially presence of a high number or high percentage of morphologically abnormal spermatozoa. And back in those days, they would refer to that as spermatoria or seminal weakness, and they would attribute that to marital excess. So how much progress have we made since then? Quite a bit. We have much better microscopes and much more expensive microscopes, but essentially we still rely on subjective light microscopic analysis, where we look at sperm count or sperm concentration in a sample, sperm movement or motility, and sperm appearance or morphology. And this allowed us to develop baselines for semen parameters in livestock species and in humans, based on which sires can be used or are either used or not entered into artificial insemination breeding programs. This conventional semen analysis, of course, provides very useful baseline information, but the issue is that it does not always necessarily reflect fertility. And the reason is simply because there are three different kinds of spermatozoa. There are the good ones, the bad ones, and the ugly ones, to paraphrase a famous spaghetti western movie. The bad and ugly spermatozoa are very easy to spot if you have a good quality high magnification light microscope. However, excuse me, let me track back. So again, they are very easy to spot, and we have been able to literally catalog and classify a number of sperm head and tail defects in livestock animals such as bulls and boars. However, sometimes the spermatozoa that are defective actually look morphologically normal. The appearances can be deceiving, and it is only through application of biomarker-based semen analysis approaches that we can actually distinguish spermatozoa that are morphologically normal but defective at the subcellular or at molecular level. So with this new type of technologies, our goal is to provide better tools for fertility assessment and develop biomarkers that will be reflective of individual fertility of a sire, and even reflective of the fertility of individual semen sample, of individual semen collection. The major advantage of these technologies is that they are objective, so they are not based on the judgment of the observer evaluating the cells under the microscopes, and they can detect a number of different anomalies that are not necessarily manifested by abnormal morphology. And these assays ideally should be low cost and should be multiplex assays that can in a single run perform or test a variety of sperm cell attributes. Over the last 2 decades, a number of such techniques have been developed. So in the next section of my talk, I would like to particularly pay attention to methods that are based on computer assisted analysis and methods that are based on the technique of flow cytometry. The introduction of computer-assisted semen analysis, or CASA, of course, have been a major advance in the field of animal and human andrology, because this technology can not only provide sperm counts and motility, sperm movement evaluation, but it can actually trace individual trajectories of spermatozoa and distinguish between very subtle changes in sperm motility that can be associated with good fertility, or actually associated with moribund dying spermatozoa that have been damaged or entered the process of premature sperm capacitation. Flow cytometry adds next layer of sensitivity to this process, as we can very quickly analyze thousands of cells in a matter of few seconds by labeling the cells with vital fluorescent markers, or with lectins and antibodies that can be used on a fixed sperm sample. These cells are fed to flow cytometer in a stream of sheet fluid at a speed of several thousand per second, and the fluorescent dyes here are excited by lasers. This excitation emission, this excited emission is then collected by photomultiplier tubes and analyzed by a computer, and the computer outputs give two types of useful information. First of all, it is the fluorescence intensity from the fluorescent dye used to mark either normal or defective cells. And then it is also a scatter diagram, which essentially for each and every cell passing through flow cytometer provides sort of a footprint of the size and to some extent the shape. The shape of the cell, each dot here being one cell. So by fluorometry, we can see when we examine a probe specific to a biomarker associated with defective sperm cells, we see a distinct shift in the peak in the fluorometry of this fluorescence histogram, and then with visible transmitted bright field light, we can see the scatter diagrams where in a normal healthy sperm sample, most of the dots, most of the cells are fairly uniform, of similar value, and they essentially are within the same focus in the center of the scatter diagram. In males with sperm defects, we often see abnormally large flow cytometric events up here in the upper right corner, and also we see cellular debris shown here because of its small size in the lower left corner. So we get two types of useful information, and this has been widely used for semen analysis, where we can use vital dyes for mitochondrial membrane potential that is reflective of sperm ability to move and highly correlated with sperm motility. We can use live dead stains to distinguish between live and dead spermatozoa and establish the percentage of dead spermatozoa. We can analyze presence of damaging reactive oxygen species in spermatozoa and in the sperm sample. We can examine chromatin structure and integrity of sperm DNA, presence of DNA strand breaks, and we can also examine the integrity of the sperm acrosomal cap, which is very important for sperm ability to fertilize the egg. One of the more recent developments in this type of technology has been focused on sperm capacitation. Capacitation is the process by which spermatozoa, after being deposited in the female reproductive tract, acquire the actual fertilizing ability, the ability to fertilize the egg. And one of the early hallmarks of sperm capacitation is the influx of calcium ions into the interior of the sperm cells. If this occurs prematurely, of course, this spermatozoa will die. And if there is a large number of prematurely capacitated spermatozoa in artificial insemination dose, this will significantly reduce or even completely eliminate fertility of that insemination dose. The problem encountered with measurements of calcium influx by flow cytometry is that oftentimes this is somewhat difficult to interpret, and we do not always get these clear, nice histograms out of using calcium reporter fluorescent dyes. A few years ago in my laboratory, we have actually discovered another ion-based early event of capacitation process, and that is actually efflux or release of zinc ions. Zinc is important for male fertility and is present in high concentration in spermatozoa and seminoplasma, and as we have discovered, those zinc ions are released at the time of capacitation. So combining this observation with flow cytometric technology, we were able to assign so-called zinc signatures to spermatozoa at different states of capacitation. The intact, non-capacitated live spermatozoa would have zinc ions in the head and tail. After capacitation, the zinc concentration would be highest in the upper part of the sperm tail, in the midpiece, and as the spermatozoa started dying off post-capacitation or might be damaged by semen processing and cryopreservation, they essentially lose zinc ions. So that's the zinc signature of dead sperm cells. And this zinc signature, of course, changes during sperm capacitation and also changes during semen processing. What it does is it actually regulates the activity of hundreds of proteins that depend on zinc that are in the spermatozoa. Many of them are enzymes, metalloproteinase type enzymes involved in fertilization process, for instance. So essentially at capacitation, the zinc efflux activates these proteins in spermatozoa in preparation for fertilization. And we have also recently found that semen processing affects the sperm zinc signature, and with particular interest into semen sexing, which is now being used more and more, we have found that the semen sexing procedure, separation of spermatozoa bearing X versus Y chromosome, actually has negative effect on zinc signature, and in some ways actually simulates premature sperm capacitation. So our efforts now go towards managing these detrimental effects of semen processing, and we are trying to do that through management of the sperm zinc signature, so we can generate artificial insemination doses with more viable and fertile spermatozoa. And we actually can even, to some extent, reverse the process of premature sperm capacitation. As I mentioned, seminal plasma has high content of zinc ions, and so by diluting semen for artificial insemination doses, we are essentially diluting out that desirable zinc. So we came out with technologies for zinc reloading, adding it back into processed semen, and with that we can increase the percentage of spermatozoa with desirable non-capacitated zinc signature, and we can actually extend the shelf life of processed extended semen, which is particularly useful in swine industry, where most of the artificial inseminations are done with fresh semen as opposed to cryopreserved semen. So the shelf life of fresh semen is of utmost importance to swine industry, and we have been developing new types of extenders based on zinc management that would actually allow fresh semen to contain live spermatozoa not only for several days but actually for several weeks of storage at either room temperature or at +4 degrees. And of course, being not only translational but basic researchers, we are interested in how zinc influences the sperm fertilizing ability, and how it regulates those sperm zincoproteins. So we have been conducting studies of the whole sperm proteome with focus on zincoproteins, and we have found a number of very interesting, previously unknown phenomena. One of them being the activation of pathway for Huntington disease occurring during sperm capacitation. This is all work in progress, so I'm not going to delve into a lot of detail on it, but it does introduce the next section of my presentation, talking about use of proteomics and various omics, in fact, for development of better technologies for semen analysis. And with this new tools, we can essentially examine the sperm transcriptome. We can focus on non-coding RNAs being present in spermatozoa that might actually be brought into oocyte, the egg at fertilization, and regulate early development of fertilized egg. And we are looking at metabolites that are produced during sperm capacitation and semen processing. And we have been doing a lot of work on proteomic analysis of normal versus abnormal sperm cells, and we can do it in different ways. We can take a sample from a fertile sire, separate the effective spermatozoa from normal spermatozoa by various types of purification procedures, and we can then examine and compare the proteomes, the protein makeup of those fractions within a fertile sample. Similarly, we can actually compare whole proteomes of sperm samples from fertile versus infertile individuals, and over years we have identified a number of different proteins that essentially have been validated as biomarkers of sperm quality and fertility. So I'm just going to very briefly mention some of our most prominent ones. One of them is ubiquitin, a protein that serves as a recycling tag for protein turnover in every cell, including the spermatogen. And by flow cytometry, we have cells and observed consistently that sires with poor fertility have an increase of ubiquitin content in spermatozoa, and that correlates with fertility. Another example, somewhat different, is this protein called WBP2NL or POP. This protein is not associated with sperm defect. To the contrary, it is important for fertilization process, and it helps with process of oocyte activation and fertilization. But in some sires, we can either see a spermatozoa that carry too much of this protein or not enough of this protein. And that again allows us to correlate the content of this biomarker in a semen sample with sire fertility. Interestingly, other groups recently discovered that the gene encoding this protein is actually expressed in several types of cancer. This is a male germline specific protein only expressed in male germline, so no somatic cells or female germ cells will express it, but it is expressed in breast cancer, and it can be used as a biomarker for breast cancer diagnostics. So finally, I would like to discuss some recent developments in technology. As I already alluded, the major advances in andrology have been brought about by technology improvement, and it was for a long time it was microscopes, but now we are actually combining the power of microscope or the resolution of microscope and the imaging ability of microscope with the high throughput and sensitivity of fluorometry in flow cytometers of new generation, which are referred to as image-based flow cytometers. So these machines can measure fluorescence intensity in individual cells at the rate of around 2,000 cells per second, generating the histograms that I told you about. But at the same time, these amazing instruments can actually photograph each cell passing through in bright field and up to 10 or 11 other fluorescent channels. So we can not only measure the fluorometry, but we can actually create galleries and galleries of individual cells and examine where a given probe is giving positive signal within the sperm head or sperm tail. Initially, this technology was developed as a research-only instrument, but now it's been scaled down for benchtop use. So just like conventional flow cytometry, it is now making inroads into clinical andrology, primarily on human side, but hopefully also on veterinary side. And this is amazingly powerful technology because by simple use of DNA stains and lectins in this example, you can see how in the same sperm sample we find not only one or two distinct separated cohorts or subpopulations, but actually we can find 5 or 10, depending on what combination of biomarkers and reporter fluorescent probes we use. We can also actually correlate this very well with the bright field images generated by this technology, and we can literally develop phase IDs of individual spermatozoa. We can essentially train computers to evaluate initially based on fluorescent reporters, but then based solely on bright field images, which sperm cells are normal or defective, regardless of whether or not those defects would be manifested by gross morphological abnormalities. So we are literally introducing AI into AI, artificial intelligence into artificial insemination, and the endpoint of this process, essentially going through biomarker discovery and validation and machine learning, is to provide applications where we use label-free approach, simply feeding cells into these smart flow cytometers and attached computers without having to use any dyes, any fluorescent probes, just to do label-free, very quick analysis. So this is where I see the very near future of andrological semen analysis, and that again brings us to the point of being able to take full benefit of advances in genomics. So now we can actually use this technology to phenotype spermatozoa from sires that carry distinct defects. We are focusing on single nucleotide polymorphism and insertions and deletions in the genes that affect and sperm structure and function. And a number of such mutations have already been reported, and this is a growing list. And also we now have thousands of sires worldwide that have been fully genotyped. And with that, it is much easier for us to focus on specific mutations. And this is an example from our lab, a recent work on a microtubule-associated protein that carries a rare mutation affecting its function. And at the phenotype level, at the sperm phenotype level, this is causing a very peculiar defect of the spermacrosomal cap with this big protruding knob. So based on this type of approach, essentially coupling genomics with advanced sperm phenotyping, we will be able to diagnose males carrying such mutations even without having to genotype them. And we will be able to essentially mitigate and possibly even completely remove such detrimental alleles from the gene pool used for artificial insemination. So finally, in the very last part, I just want to show you how this knowledge can also let us improve individual artificial insemination dose, and that is by getting rid of both the bad and the ugly spermatozoa through semen purification process. And there is multiple benefits to semen purification, because it improves male fertility and increases pregnancy rate after artificial insemination. And it may also allow us to actually reduce concentration of sperm for semen dose, so that spermatozoa from sires with high genetic value in production traits can actually be used for inseminating more females. And by removing this defective spermatozoa, we are also removing important source of reactive oxygen species that within the semen sample will have negative effect on the normal spermatozoa. So currently there are various types of mostly gradient or matrix-based technologies for semen purification. But in our technology, we take the advantage of our biomarker validation studies, and we focus on sperm surface antigens and ligands on the sperm surface that are associated only with defective spermatozoa, and we use lectins and antibodies that essentially can be attached to nanoparticles, particles smaller than 100 nanometers. And these particles can be simply mixed with semen during processing of the dose for artificial insemination. And since these particles are made of iron oxide, they can be magnetized. And once these particles bound to the spermatozoa, defective spermatozoa in a test tube, we can simply use a strong magnet, no need for centrifugation, which by itself is damaging to spermatozoa, and we can simply bring the defective spermatozoa down to the pellet at the bottom of the tube. This is the strong magnet bar used for such process, and this is the pellet of defective spermatozoa and remnants of the dead spermatozoa, cellular debris that accumulates at the bottom of the tube. This is what that sample looks like after purification. So initially we have developed this technology for cattle, and we have field tested that in collaboration with the U.S. Department of Agriculture. And since then, other groups have adapted this technology for semen purification in boar. Most recently, a group in Spain reported successful use in a rare breed of donkeys, and most recently this has also been used for purification of camel spermatozoa. Importantly, and going back to our notion of one health and one medicine, this technology has been adapted for use in human infertility clinics and has been used to prepare spermatozoa before they are selected, one cell at a time, for treatment by intracytoplasmic sperm injection. So to summarize my talk, I would like to once again stress out the importance of biomarker-based approaches for semen analysis and the ability to reveal subtle, subcellular, and molecular sperm defects that would not be detected by conventional light microscopy-based semen analysis. And I also want to emphasize the importance of new types of technologies such as omics, nanotechnology, and improvements of artificial insemination that altogether are leading the way towards increasing reproductive efficiency and reproductive health in livestock, but also in human. And we have been applying some of these technologies to our joint project with our friends at your university, looking at yet another important agricultural species, livestock species, and that's the ostrich. And it's really a fascinating study, and I'm very grateful to John and Lizette that they made all this possible by starting this wonderful collaboration. That is ostrich cells and the ostrich testes, as you can see it here on a cross section of seminiferous tubules, are truly some of the most beautiful germ cells or most beautiful cells of any kind I have ever seen in my career, and it's been truly a joy to collaborate. And in the last 2 years, we've been mostly doing that remotely, but hopefully we will soon be able to pick up where we left off and just visit. So with that, I would like to thank not only my collaborators and all of you, but also briefly acknowledge our funders, U.S. Department of Agriculture and the National Institute of Health, as well as the University of Missouri. And also I would like to acknowledge my laboratory. We've been at the University of Missouri for 20 years now. Miriam and I started this laboratory together, and Miriam, my wife, is also my lab manager and the research technician, and together we have been working with a number of very talented students and associates in the past and in the present, and they are the ones responsible for all the beautiful pictures and all the beautiful data that I have been honored to present. So thank you one more time. It's an incredible honor for me to present this lecture, and it is very much appreciated. If I can be of any assistance, please make sure to find me, email me, visit our lab website. Thank you so much.
Thank you very much, Professor Sutovsky. To us, being isolated at the southern tip of a very large continent, on the what we like to call the right side of the equator, but where most of the scientific activity takes place at the other side of the equator, it is of extreme importance to us. In fact, it is vital that we have collaboration and links with our colleagues far afield. So thank you very much. Your contribution is highly, highly appreciated. We also acknowledge our speakers by presenting them with a certificate, and this will be mailed to you. But with this certificate, we honor an esteemed colleague, scientist, and researcher for his outstanding contribution to the promotion of science. Thank you very much.
Thank you. Thank you. Thank you so much.